My name is Blaise Kimmel, and I'm a professor here at Ohio State. My background is entirely in chemical engineering, and I did my undergraduate here at Ohio State before I went on to do my PhD at Northwestern University. I recently finished my postdoctoral research fellowship at Vanderbilt University, and I'm excited to be back at Ohio State so that we can study some exciting protein immunoengineering research. In my lab, we are fully focused on the idea that we can modulate and tune the immune system. Now, in order to do that, our group considers a couple of fundamental ideas. We have expertise in the field of protein engineering. Now, protein engineering is an exciting area because in that realm, we're able to actually utilize bacteria and mammalian cells in order to create unique proteins. The proteins that we're really interested in are called antibodies. Antibodies have this really interesting shape like a Y. And what's really exciting about them is they have really good specificity of two different regions. The region here at the top, which is based on both of these sides, is actually able to bind onto bad things. Now, these can specifically be used in a way that can help you to bind really effectively onto pathogens of interest, receptors that exist on previous existing cells in your body, but also onto new types of novel targets that we use in cancer immunotherapy. This makes it called the Fab region. At the other side of this, we have what's called the FC region. Now, this is able to bind onto immune cells in your body and help to recruit them into the site of where might be a cancerous cell bound by the Fab region. Now, traditionally, people can use protein engineering for a lot of different advancements in delivering proteins that might be lacking in a cell, or to help to modulate the immune system based on the interaction and communication from one cell to another. Another area that we study in my group is actually an area called medicinal chemistry. Now, medicinal chemistry is exciting and really takes advantage of the research I did in my PhD. Medicinal chemistry means that we can come up with exciting new drugs that have the ability to help to activate or suppress different elements of your body. In our group, we're thinking about agonists and antagonists, and these are molecules that are able to actually perform that function with the immune system. When we combine both of these together, we can consider ways to load these drugs onto an antibody. The antibody then helps it to improve the half-life of the drug, and it also makes sure that it gets to the right place at the right time. But in order to do so, we need to link these two together, and that is a huge element at which my group is able to function. By combining a linkage chemistry that takes an antibody and a drug together and making that linkage very precise, what we can do is actually create unique constructs that allow us to deliver novel medicinal chemistry drugs to create exciting new immunotherapies. And together, what this allows us to do at Ohio State is to venture into a world of cancer immunoengineering. Of that, I'm excited to play a deep role. We have partnerships and collaborations here at Ohio State between the Cancer Center and the College of Engineering, and together through this, we can see this as a unique cycle that we can be creating exciting new innovative drugs and therapies for the use here at Ohio State. I hope that you find this exciting, and I hope that you consider working with our group together. We would be happy to take undergraduates, graduate students, and postdocs, and we look forward to all the adventures we're going to have here at Ohio State.